and welcome to the next lesson of Advanced Higher Biology, which is about the synthesis of membrane proteins. So before now we have been looking at intracellular membranes and the fact that within the cell we need a series of multiple membranes, both the external plasma membrane surrounding the cell and we need a membrane surrounding each one of the individual organelles, and that allows an increased surface area on which the different reactions that are going to take place can occur, allowing the cell enough space for those reactions to happen. What we're going to look at today is the synthesis of the membrane proteins, and we will also have a wee look at the other parts of the membrane as well and how they are synthesized. After that, we're going to move on to looking at how those proteins are moved either within the cell or secreted out with the cell. So we're still looking at the process by which cells make proteins. And after this, you need to be able to state the location of both lipid and protein synthesis, state what is different about cytosolic proteins, and explain what happens during the synthesis of transmembrane proteins. So a quick recap of the structure of the plasma membrane. So you might remember from National 5, we didn't really cover this in much detail at higher, that the plasma membrane is referred to as the fluid mosaic structure, meaning that it is made up of these phospholipid molecules which form a bilayer, and it has associated proteins stuck in with it. These phospholipids are made up of a phosphate head with fatty acid tails, the head being hydrophilic or water loving, and the tails being hydrophobic or water hating. And that means that they orientate themselves so that the head is facing out the way and the tails are facing in the way, and that's what gives us this bilayer formation. The plasma membrane also has proteins associated with it, and those proteins are going to be dependent on the type of role that they need to carry out and the type of role that whatever the plasma membrane is surrounding needs to carry out. If this is the external plasma membrane of the cell, or if it's the plasma membrane that is surrounding one of the organelles. To begin with, we'll have a little look at how these phospholipids are synthesized, and this is going to take place. We're going to be having a look at a new structure of the cell, which is something called the cytosol. Now, really, the cytosol is just our new term for the cytoplasm, which you will already have heard about at National 5 and at a higher level. So the cytoplasm, or cytosol, is our jelly-like liquid which is surrounding all of the organelles and which contains some of the chemicals for different reactions to take place. And we're now just referring to that from now on as the cytosol. So within the cytosol itself, we have the precursors for the different parts that make up the plasma membrane, that make up specifically the phospholipids. So those phosphates and lipids are floating around within the cytosol and is actually the endoplasmic reticulum, this organelle here, which is going to be involved in their synthesis. When we look at the endoplasmic reticulum, remember it is a network of membrane tubules which are surrounding the nucleus. So if this is our nucleus around here, then the endoplasmic reticulum is these tubes that are surrounding it. And we've got two forms of endoplasmic reticulum. We've got smooth endoplasmic reticulum and rough endoplasmic reticulum. And rough is just that which has got ribosomes present on it. Now, when the smooth endoplasmic reticulum is making these phospholipid molecules, what it's doing is it's taking the smooth endoplasmic reticulum, it's taking those precursors for the lipids, and enzymes that are present on it are starting to build them into the bilayer. And what it does is it just extends its own membrane by doing that. So it's just making new membrane by extending its own. And over here you can see where it's making an extension out in this bit here. We're building a new section of membrane over here. If that membrane needs to remain in the smooth endoplasmic reticulum, then fantastic, but that's not usually the case. Normally we're producing new membrane for another organelle or another part of the cell. It might be because the cell needs to grow and expand, so we need to make our membrane slightly bigger. If that's the case, then this section of the smooth endoplasmic reticulum, this new section of membrane, is going to form a little bud, and that's going to disappear off into the cytosol as a transport vesicle. So this vesicle here is just membrane, that's all it is. And that's going to float off and to be taken to whichever organelle it needs to go to, or to be taken to the external plasma membrane of the cell. Proteins are a little bit different. Now, you'll remember from National 5 and higher that proteins are produced in the ribosomes, and that's not any different now. But what happens to them might be a little bit different from what we've previously known about. So, all proteins are begun in the cytosol. Ribosomes can either be floating free in the cytosol or can be attached to our endoplasmic reticulum to make rough endoplasmic reticulum. However, all protein synthesis starts at the ribosomes, which are floating free in the cytosol. And you might remember from higher that a ribosome is made up of a small and a large subunit, which are forming together, kind of locking together around our mRNA molecule here. That's not particularly important to know about at this time, but it just helps explain this diagram a little bit. So if we've got our ribosome and it's attached onto the mRNA molecule, it's going to be reading along it and producing its polypeptide. So the polypeptides that are produced might need to remain in the cytosol, they might need to be passed into other organelles, they might be proteins which are making up part of a plasma membrane somewhere, or they might be secreted from the cell to be used elsewhere in the organism. And regardless, the ribosome is going to start their translation in the cytosol. However, how does it know where they're going to go afterwards? Essentially, all proteins, all of these polypeptide molecules, carry a short signal sequence, and that is recognised by the ribosome, and it's going to direct a complete polypeptide into the correct location. So there's got a short section of amino acids along here, and the ribosome recognises that and is able to tell the polypeptide where it's going to go. And there's really two main options at this point, once it's recognised that signal sequence. If the protein is a cytosolic protein, so it's one that's going to remain in the cytosol, then the ribosome is going to complete its translation of that polypeptide within the cytosol. It's not going to go anywhere. It stays where it is and it carries on producing our completed polypeptide. Now, chemists among you and other particularly eagle-eyed individuals will have spotted these particular chemical groups at either end of our polypeptide. Don't worry about those for the moment. Yes, they exist. Yes, we will need to look at them, but we'll be looking at them next week when we consider the actual structure of a protein itself. For now, just know that this is our completed polypeptide. It's been produced by our ribosome. Our ribosome is now disbanded and left its completed polypeptide in the cytosol. So from there, it may remain in the cytosol, or it might be imported into the interior of an organelle to be used there. Our other option is if we have a transmembrane or an external protein. So in this case, our signal sequence is going to be recognised by a ribosome, and that prompts the ribosome to bring the polypeptide over. It brings it all the way over to dock with the endoplasmic reticulum. So the ribosome sticks onto the endoplasmic reticulum, forming rough ER. From there, it continues its translation, and when it's finished, again the ribosome disbands, and we have the completed polypeptide remaining in the endoplasmic reticulum. If that protein is to form part of a membrane protein, it will remain in the endoplasmic reticulum and be built into the membrane as the endoplasmic reticulum is making that new membrane that can then bud off and be taken elsewhere. Otherwise, it will be taken into the Golgi apparatus, where it is packaged up. Other molecules might be attached to it. We may have some cleavage, some other post-translational modifications might occur in the Golgi apparatus, and then it might be taken to a secretory vesicle, such as a vesicle or a lysosome, or be taken to the external plasma membrane of the cell, where it will be taken away and secreted to another part of the organism for use by a different cell somewhere else. So quick recap, looking at the location of lipid and protein synthesis. Lipids are made on the smooth endoplasmic reticulum from the precursors that are floating free in the cytosol. Proteins are begun in the ribosome in the cytosol, so they're begun on cytosolic ribosomes, and then depending on their eventual location, different things are going to happen to them. If it's a cytosolic protein, then that ribosome will remain in the cytosol, where it's going to complete its translation of the polypeptide and leave the protein floating free in the cytosol. If it's a transmembrane or an external protein, then the ribosome will be redirected to come and dock with the endoplasmic reticulum, and the completed polypeptide will remain in the endoplasmic reticulum and be built in, along with these lipids, into new proteins, or it will be passed on to the Golgi apparatus, where it can then be packaged up and secreted out with the cell. I hope all of that's made sense to you. As ever, please feel free to ask any questions as we're going over this in class, and we'll be making some notes on this.